Okay, today I am going to teach you about the molecular markers. The molecular markers are the DNA fragments which we use to identify the genetic disorders or something else based on the genetics, and these DNA fragments are called as molecular markers. So we can define the molecular markers, which are also called as genetic markers, as the variant DNA fragments that give some information about the gene of interest are the molecular markers. The important molecular marker techniques include RFLP, RAPD, AFLP, VNTRs, and SSR, and many more. But we are going to discuss here according to the syllabus. So, first technique, that is RFLP, I am going to discuss it here. So, you can mark your heading RFLP. The full form of RFLP is restriction fragment length polymorphism. Uh, Suppose there are two individuals. I'm going to explain you the concept and the technique for RFLP. So suppose there are two individuals. One of the individual is a normal individual, and another individual is an individual which has some genetic disorder, maybe L-captonuria, phenylketonuria, or some other genetic disorder is there. Genetic disorder means the gene is malfunctioning and then gene is defected. Because of that defect, the disorder exists in individual 2. So in RFLP technique, we take the two individuals, the DNA of the two individuals, we take the cells of the first individual, we take the cells of the second individual, we isolate the genomic DNA of the first individual, we isolate the genomic DNA of the second individual. Then we cut these two genomic DNAs with certain restriction endonuclease. So for example, in this case, we have taken EcoR1, but in both the cases, the same restriction endonuclease has to be taken. So this is the first step, that is the restriction digestion. So we isolate the genetic DNA by the standard techniques that I am not going to discuss here. Then these DNA, both the DNAs are cleaved by restriction endonuclease, that is EcoR1 or some other restriction enzyme, and then these restriction fragments formed are called as restriction fragments or restriction DNA fragments or restriction fragments. Now these restriction fragments will be of different length because these two the DNAs or gene will be different. Okay, so we say the polymorphism exists in the restriction fragment because uh, same endonuclease cuts the DNA giving different lengths of the restriction enzymes and different lengths of the DNA. That's why we call this technique as restriction fragment length polymorphism. So next what we do here, these DNA fragments are then gel electrophorized. Their single strands are electrophorized. So we make the single strand out of this double strand, and one strand we uh, separate for making the probes. Now, during gel electrophoresis, we take a gel electrophoresis tray, and in two different wells, we release the first DNA fragments and the second individual DNA fragments separately, and these two DNA fragments are never mixed together. So the DNA will be separated here because of the electric current, and this is called what we say gel electrophoresis is occurring here. And when these DNA fragments are separated, they are subjected to Southern blotting. During Southern blotting, we transfer the DNA bands of both the DNA in the nitrocellulose paper. So these are the gel, the DNA which we have taken here, the nitrocellulose paper is there, and then a bunch of blotting paper is there, and then weight is there. When we apply the weight, the liquid which is present on the gel will move up, so it will raise the DNA bands from the gel onto the under surface of the nitrocellulose paper. We separate this paper, and this technique is called as blotting technique, that is Southern blotting. And then we separate out the nitrocellulose paper, and then we add the radio label probes. The radio label probes are the certain nucleotide sequences which are been inserted with P32 radio labeled phosphate, and then these bands will release radiations, and then radiations are taken on the X-ray film, and the bands are developed. So when these radioactive probes are released on the DNA bands, they base pair according to the base pair rule with both the DNAs. Then what will happen? These radioactive probes hybridize with the DNA molecules of individual 1 and individual 2, and this technique is called as nucleic acid hybridization. So after nucleic acid hybridization, the radiations will come from the lower DNA, that is the radioactive probe, and they are taken on the X-ray film. These X-ray film, through these X-ray films, we can analyze the nucleotide sequence of the first DNA and also the nucleotide sequence of the second DNA, and this technique is called as autoradiography. And then both the DNAs, the nucleic acid DNA of sequence of the first DNA is compared to the second DNA, and by comparison we can analyze the defect. Where lies the defect in the second DNA, because the second DNA is showing genetic disorder, and this is how we can compare the two DNA. So it's a comparing. These DNA bands are used. These DNA bands are used for the comparison of the normal and the defective DNA, and these DNA bands are because developed by the restriction uh, enzymes, and that's why the technique is called as restriction fragment length polymorphism. So that's all for this topic. I hope the topic and concept is clear to you, and so I'm going for a quick revision of what we have done in RFLP. We take the DNA of the two, we can take the whole genome or the particular gene of the two DNA of the two individuals. These two genes are then cut with restriction enzymes, and the restriction fragments are developed. These restriction fragments are separated through gel electrophoresis. Then they are taken on the nitrocellulose paper, and this technique is called as blotting technique. And then nucleic acid hybridization is done by radioactive probes, and then by autoradiography we can find out the nucleotide sequence of the first DNA and the second DNA, and then both the DNAs, that means both the genes are compared for the defect. And this is what we'll do, what we exactly do in RFLP. Hope the concept is clear to you. Thank you.